Start. Okay. Good morning to all. Jai Hind. Myself, Dr. Ganesh Mishra. On behalf of Faculty of Pharmacy, Swami Vivekananda Subharti University, Meerut, welcome all of you. Respected Dr. Sukender Kumar, Principal and Dean, Kharwal Subharti College of Pharmacy. Today's speaker, Dr. Sachi Bharadwaj, Application Scientist, Altem Technology Private Limited, India, faculty members, and dear students. On today's webinar entitled Advances in Computer-Aided Drug Design from Algorithm to Therapeutics. Now, I would like to request Dr. Sukender Kumar, Dean Faculty of Pharmacy, to say a few words about today's webinar.
Dr. Ganesh, am I audible? Dr. Ganesh, am I audible?
Yes, sir, you are audible.
Okay. Good morning and Jai Hind to all. Today webinar is titled, as Dr. Ganesh told about
Hello. Not audible,
Hello?
sir.
Yes, sir. So I again start. Good morning and Jai Hind. First of all, I welcome our esteemed expert, Dr. Sakshi Bharadwaj, who has given her consent and she is ready to deliver her expert lecture. So first, it is my privilege and honor to welcome our esteemed speaker and guest. And from the behalf of the Faculty of Pharmacy, Swami Vivekananda Subharti University, I welcome all the participants. And I want to tell you about the drug discovery, right? Drug discovery is an ongoing process. Every time we discover and develop the drugs, we need large number of drugs to treat, to mitigate the disease. But this is actually the traditional drug discovery process is a lengthy or very cost-effective process. So recently, in few years, the CAD, computer-aided drug designing, have played a great role to reduce the cost, to reduce the time, and to make the precise drug. So today we will welcome our speaker, Dr. Sakshi Bajaj. She will give a expert talk. Now I over to Dr. Ganesh Mishra, please.
Okay, sir. Now I would like to introduce today's speaker, Dr. Sachi Bharadwaj, Application Scientist, Life Science LM Technologies Private Limited, India. She has more than 11 years of experience in computational approaches of drug design like QSAR, pharmacophore, and homology modeling, structure-based drug design, ligand-based drug design, and molecular simulation. Apart from this, she has expertise in instrumentation techniques like UV, IR, NMR, and chromatography techniques. Now, I request Dr. Sachi Bharadwaj to start your presentation.
Thank you, sir. Thank you for the introduction. Myself, Dr. Sakshi Bharadwaj, Application Scientist, Alton Technologies, Bangalore. First of all, I would like to thank you to all of you for joining this session, which is all about going to, for advancement in the computational drug designing with the algorithms to the therapeutics. How those algorithms, how we can utilize those algorithms to get one drug kind candidates. Okay, it refers to the process basically in the drug discovery and the development in the field of drug designing with the help of computational methods and the techniques to assist or could be helpful in the discovery and designing of the new pharmaceutical candidates. Actually, this field sits at the interface of computational science, chemistry, and the biology, aiming to speed up the drug discovery process and reduce the time and the cost associated with bringing those novel candidates or the drug to the market. Okay, before starting, I would like to introduce my company's profile to you all. We are leading company in the 3D innovation, and we have various wings in the science center technologies, where we have life sciences with us, where we are using the bioway discovery studio. And here we are company those using the 3D innovation platform, which I would say that could be the solution for all of your problems. Starting with my slides, actually, I will take very brief introduction about what is a drug designing. We have heard a lot of terminology like drug design, in silico drug design, CAD, rational drug designing. So what all these terminologies are? Let me quickly refresh a drug design concept which based on the knowledge of biological, targets, basically finding a new medication, which what we call as a drug design. Okay, actually these all are associated with the chemical component, whether it is from the macro molecules or from the small molecules. The goal seems to be same: the development of a new candidate in the process of this drug designing and discovery process, okay, which can interact with the specific target macromolecules, that is your biomolecules, okay, with some kind of interaction, so that we can get a therapeutic component or a candidate which could be a drug like candidate, and hence we can bring that candidate to the market for the formulation and further for the after these DNA approvals kind of things. In silico is nothing but, as we are going to use the computational tools here, so and of course the computers are using those silicon chips. That's why it is called as a in silico drug designing, okay, which comes from a Latin word that is the silicio. Okay, coming forward, you can see here the two ways of drug discovery. One is the conventional method where we are using our chemical labs. The other way is the in silico drug designing, where we are going to use the computational tools or the techniques. Okay, when it comes to the conventional wet lab, it involves actually the physical experimentation with the chemical components and the biological system, which require extensive resources. Okay. And of course, it requires your time, your efforts, which based on the hit and trial and error methods, right? Here we need to create some kind of chemical libraries, okay, for our small molecules, which we call as a computational libraries with the synthetic process. And then we can screen these libraries against a particular biological target to identify the potential leads, of course. Okay, these compounds interaction with the biological target, actually uh, we need to cross all those pharmacokinetics, toxicity parameters. When it comes to the wet lab, we are doing those toxicity prediction or the EDMAT prediction or the pharmacokinetic stability or pharmacochemical stability experiment in the wet lab itself. Which so basically this approach requires access to the well-equipped libraries for the chemical synthesis, purification, and for, of course, the biological testing. But when we comes towards the in silico drug designing, it involves the use of computational techniques, basically to model and predict the interaction between the molecules and the biological system. Then only we can identify, okay, this particular drug is going and binding to macromolecules, whether in terms of activity or deactivity, whether it will stimulate or inhibit that particular kind of macromolecule. Okay, we can do some kind of virtual screening for our large libraries. We need not to go for the conventional method to have a chemical component in our hand. We can do the virtual screening with help of these computer techniques by saving our times and resources. Okay, So here, based on the computational predictions, researchers can choose those molecules to synthesize in an informed manner. When I say it's an informed manner, that means we have all the information about that particular chemical component. Hence, this could be a rational approach. That's why it is called as a rational, because we are making ourselves sure while getting that particular chemical component, which could be a drug-like candidate. So here are different terminology involving like hit molecule, lead molecule. So nothing to worry about these terminology. Hit molecule is nothing but a hit candidates from all those 10,000 or lakhs of molecules. Those are going to lead the series. Okay, like a leader, for example. Okay, and hit molecule is what? Among those hit molecules, that particular hit molecule is going to be a drug-like candidate. Okay, so hence we can utilize these computational methods, okay, for their molecular interaction, binding affinity, EDMET predictions before any experimental work, right? So that's why we are using computational tools basically to mimic the biological system here. We rely on the power of your computers, your softwares, and of course, softwares. To run those softwares, we need some kind of powerful algorithms for the various predictions like virtual screening, lead optimization, target identification, molecular modeling, docking, simulation, and of course, further for the analysis process, right? But ultimately, after using that in silico drug designing, computational technique, also we need a wet lab to design our that particular hit molecule, rather than we are designing a series of ligand molecules. With the help of in silico prediction, we are getting, or we can optimize, I would say, we can optimize those hit molecules to get one kind of a hit molecule, right? So that is the main difference in between the hit molecule in the in silico drug designing and the conventional wet lab. This slide is very common. Actually, you can see this picture anywhere. Okay, but it is very important. Try to explain the drug discovery and development process, which takes 10 to minimum 10 to 15 years actually, starting from the identification of your disease condition, then the isolation of protein, then the preclinical or the identification of your target molecule, lead molecule, that could be your chemical structure, and then further for the preclinical testings. Of course, we need some scale up or the formulation of that particular hit molecule to be enhanced. Then further for the different various approvals like IND, NDA, right, FDA approvals to get that particular candidate to the market. Okay, but we have to face various complexity associated with this drug discovery, starting with the selection of that particular candidate, whether it is going and binding to the target site or not. It could be potent in terms of efficacy or the affinity. It should have that physical stability. It should have that proper solubility, permeability. It should be metabolic stable, whether it is reversible or not. It should be have that durability criteria or the parameter with it. It should not go for any kind of toxicity. And of course, it is patentable and manufacturable, right? Now, when we know that there are so many drugs are already available in the market, then what is the need of development of novel candidates? We all heard about the development or drug designing of the novel candidates. For example, if I'm taking the tuberculosis example, right? We know that there are so many first, there are many isoniazid like ethambutol. So many candidates are already available in the market. Hence, we are in the research of getting the more potent or more efficacious, I would say, in terms of bioavailability candidate to for against that particular any kind of a disease condition. Actually, the problem associated with the existing drugs like MDRA, XDRA, TDR, what these are, MDR is the multi-drug resistance. We all heard about this terminology, but we don't know what exactly MDR is doing. MDR is a multi-drug resistance where the multi drugs are not properly, I would say, not properly going to the side and show those kind of interaction or whether in terms of activity or the interaction. Then XDR, that is the extended drug resistance, total drug resistance. Now here the term comes, the total drug resistance, where no drug will go and show any kind of affinity, right? Of course, those drugs which are already available in the market having some kind of adverse effect, mainly the liver toxicity, poor patient compliance, right? High cost is associated with the development of those effective drugs, not with the drug condition, not with the drugs is as such, but we have some kind of problems associated with the conventional drug systems, like tablets, capsules, granules, ointments, syrups, and the suppositories, in terms of their poor absorption, poor bioavailability, high fast metabolism, fluctuation in the plasma drug level. So all those kind of problems associated with the existing drugs. Hence, there is a need for the development of either a candidate or to identify a target which could not go and show these kind of toxic behaviors, right? Of course, pharmaceutical companies need to sustain in the market. That's why they need to, have those massive contribution towards the earnings. And of course, we need to keep eye on our patent expiry dates as well. Okay. So what could be the solution when I'm telling these complexity and the problems associated with the with the marketed drug already available in the market? So what could be the solution? As you can see, I have mentioned the in vitro and the in vivo as well. In bracket, I have mentioned in life and in glass. So even the students or many people are not very clear about this terminology, where we can use this in vitro term when we are using some lab work, and whereas in vivo when we are using this into the living organism, right? And in silico means it's a virtual one, where we are going to use the computational techniques basically. Okay, so how we can utilize those computational, as I told, those complexities. So what could be the solution for problems associated with the complexity and the problems, right? How we can overcome these challenges for what we already have in our hands, right? So I would say that in silico methods that offer a promising solution to the whether those are in terms of complexity and the challenges associated with the drug discovery process, where we can utilize the computational tools basically to stimulate, we can predict those simulations, right? We can develop those models, we can generate those pharmacophores, we can predict various aspects of drug development while using these in silico or the computer aided drug designing. Hence, these methods basically helps to streamline and enhance the efficiency of the process while reducing the cost, of course, and minimizing the risk to be using the chemicals into the wet lab. Okay. As you can see here, that this is a very basic workflow for the computer-aided drug designing, where we can, download the libraries from various databases. Either way, we can, when we have drug molecule or small molecule with us from the different databases, or we can generate, their pharmacophores. Once we have those candidates with us, then we can go for the virtual screening of our data. We can apply a variety of filters in terms of ADMET and the top cat filters, right, for their toxicity and absorption, distribution, metabolism, and excretion. Okay, hence their biological activity we can predict. With the help of docking interactions, we can filter out the good posers and the bad posers, and we can filter out the good molecules and the bad ones, right, to get that particular hit molecules, right? So how this computational techniques, how these techniques are impacting the process of drug discovery? As I explained, once we have identified a disease condition, okay, uh, various approaches those approaches, uh, once we have identified, then we have to isolate our protein structure. From where we can identify or isolate our protein structure, we have genomics, proteomics, transcriptomics, where these terminologies are come into the picture. Okay, we can do the high throughput screening of lakhs of molecules to proceed further for the virtual screening. We can rapidly produce the vast number of compounds or the small molecules with these combinatorial chemistry. We can do the molecular modeling even with the one kind of component with us for to make it better potency and less toxicity with the help of computer graphics or the models to help with the improved candidate basically. Okay, so we can easily do these kind of studies, we called it as in vitro preclinical studies, with the help of computational without going into the wet lab, right? This is a reference slide actually for what could be the macromolecules. So if I'm saying the macromolecules, those could be your proteins, lipids, carbohydrates, DNAs, and the RNAs, which are having these components like amino acids, fatty acids, sugar residues, and the nucleobases, basically, right? But now, though, it's like we are using the DNAs, RNAs, even a single nucleotide kind of research we are getting into it. But still, the protein is the preferred one. Why the protein is the preferred one as a target molecule? Because those protein structures are very specific. Okay, they behave like a lock and key module, right? So when we have those kind of specificity for with respect to your macromolecules, it's very easy for us to understand their behavior. Okay, and of course to get into the desired kind of activity, right? And of course we can identify the binding site from these protein as well. Okay, so you can see the graph here, the majority of percentage will go with the receptor as compared to the enzymes, hormones, DNAs, nucleic receptors, even the ion channels, and rest of them are still unknown. We have to identify those ones. So coming to the drug designing concept, it is which is basically based on the protein structure or as well as with your ligand structure. But what if we are having your ligand molecule as known, and if I am known to my protein structure, then it is called as a structure-based drug designing. Okay, but when the ligand molecule is unknown and the protein structure is known, we call it basically we need to start our drug designing concept from the scratch. So we called it as a drug design, de novo drug designing. Okay, but when the ligand molecule is known and protein structure is unknown, then we are going to utilize the ligand structures. Okay, based on their structural features, we are going to develop the model like OSAR, pharmacophoric models, based on their alignments, what kind of features are present in those components which are contributing towards the pharmacological activity, basically. Okay, so but when we don't know about the ligand structure and even the protein structure, then of course we have to go through the literature search, so reports, analysis, designing. We need to design our ligand molecules. Hence, we can utilize a diversity by generation of these ligand molecules. And hence, once you have any one of the candidate molecule with us, we can proceed for the further drug designing. Right. So as I told you that we are doing all work with the help of computational techniques and the tools. Okay, of course, we need some kind of components to do those kind of works, right? We need some kind of tools, we need some kind of algorithms, we need some kind of functionality, or I would say the scoring function, right? Why we need all these when we are doing these in silico prediction? Okay, very simple, an easy way to answer is like we are going to mimic the biological system, right? When it comes to the protein of to check the any kind of activity of a protein structure or a macromolecule, whatever DNA, RNA, and the proteins, right? Now, when it comes to the mimicking of a biological system with respect to with your computational techniques, right? So, in biological system, everything is perfect, right? We are taking some kind of medication; it is going to some targeted area, and it will show us the result. But when it comes to the computational techniques or the prediction, we need some those kind of searching algorithms. We need those kind of parameters to need to be fixed. That's why I will show you. That's why we need to prepare our protein structure, our ligand structure, those ligand and protein structure. Why we are preparing? The only reason is because we are going to ensure that sure that okay, if I'm taking that particular protein structure that is good enough without any false condition, I can take that protein structure and go further for my docking as well as for simulation studies, right? So of course, to predict the best model, to find out the best conformation of a ligand and protein structure, to do the rigid and the flexible docking, we both have the rigid and flexible docking biodiscovery studio, and further molecular dynamic simulation. Scoring function, why we need, because we need to select or we need to screen out all those good poses with respect to the complex ligand complex, and the way we, of course, the top one we need to take further for our simulation studies, because we consider those poses on the basis of some kind of functions, that okay, these particular ligand molecule with respect to a against a particular protein structure is showing some kind of good binding interaction. So that's why we need some kind of a scoring function so that we can rank out all those ligand molecules, right? So this is the basic slide which cover how many type, I mean, so what are the steps involved in the in silico prediction. As I told, there are two parts basically. One is related to the macromolecule, other one is related to the lead or your ligand molecules, right? When the structure of your ligand molecule is macromolecule structure is known to you, okay, we have various databases like pdb, uniprot, and cbi. Then here we can take all those structures, 3D structure. Okay, because we need that 3D structure to do those kind of to check those kind of interaction when it comes to the in silico prediction, right? But what if you don't have those 3D structure? Then homology modelings come into the picture, right? We need to identify all those faster sequences. We need to align with the help of blast search and the blast. We are doing those alignments. Then we need to go for the energy minimization processes. Of course, the validation is most important process step in each and every step here. Okay, of course, then we can identify the cavity of our 3D structure. Second, on the other side, we have the lead molecule. When we have the lead molecule or the ligand molecule, is basically called as a drug-based drug designing, where we can identify or we can check for their SAR, that is the structure activity relationship, or the qualitative structure active relationship, right? We can go for the ADMET, we can generate the combinatorial chemistries, we can develop the pharmacophoric model, and we can utilize those pharmacophoric features for further development of our candidates, which could be a active molecule or a good candidate, or which could be a drug-like candidate. And of course, once we have identified or optimized all these two molecules, we need some kind of a software to do those kind of interaction studies and the molecular dynamic simulations for us, right? So here, these are two, three, four, this web databases for the target identification, as I mentioned earlier. Whereas these are some ligand identification databases like Binding DVD, Campbell, PubChem, even Zinc, then the PDB bind as well. Okay, so actually this terminology is little confusing. What is virtual screening technique? What is high throughput screening technique? Let me make you very clear. These are all similar to one type. That is, we are going to mimic our data. That is the huge one. For example, I'm having lakhs of molecule in my series. Okay, first step what we call is a high throughput screening. Okay, then we need what we have cut down our molecule on the basis of some kind of filters. Okay, we have selected that we are using those as for the virtual screening process. Okay, further for the docking and for the molecular dynamic. Okay, because we are using everything here virtually with the help of computer, that's why those are called as a virtual screening techniques, right? You can see here the three various implementation we can do go for the high throughput screening, like as I told you, on the basis of ligand structure, development of their pharmacophore to get that one hit molecule, right? Then based on their pharmacological predictions, like when we have ligand molecules with us, we can go for their ADMET and their TopCat studies. Then we have various models. TopCat, we have various toxicity prediction models, rat, mouse, even we have some aquatic models. I will show you in my upcoming slides to get that particular hit molecule. The other way around, we have the structure-based drug designing, where we can utilize the binding site or that particular cavity of a protein where a ligand is bound, right? So by using that particular binding site, we can generate our molecules with the, to make sure that okay, this particular ligand molecule is showing better interaction as compared to those lead molecules. That could be my hit molecule. So majorly these are the three ligand molecules that give you pharmacodynamic properties on the basis of their efficacy and the potency, whereas physiochemical properties where the Lipinski rule comes into the picture. And of course, we have verbose tour in our discovery studio to reverse those physiochemical property predictions to check their chemical stability. Okay, when it comes to the pharmacokinetic properties, we have metabolic, we are going to check their metabolic stability, their toxicity predictions, right, to get that particular lead molecule. Now, when it comes to the pharmacokinetic prediction with the help of BioWay Discovery Studio, we have these 5 to 6 parameters to fix out. Right, one is the aqueous solubility, as it comes with the absorption, distribution, metabolism, and of course the toxicity. Then we are first in list is aqueous solubility. Of course, my drug will show good bioavailability, hence it must be having that particular solubility, right? So in discovery studio we have fixed at this temperature at 25 degrees centigrade to make it a neutral out. In biodiscovery studio we have kept some kind of levels. Okay, why levels? Actually, when we are checking that it is false, right? When it is false, then we are going to know, okay, it's true or false, right? So it is little easy for me to understand the results. But when it comes into the decimal values like 0.002, 0.01, 0.005, so it's a little confusing for me to identify those hit molecules. That's why we have kept the particular levels. Okay, if it is already showing level 3, that means I am taking it as a good. Then coming to the blood-brain barriers, what these blood-brain barriers actually, this users specify, if you're looking for a CNS kind of drug, then you must keep these barriers as true. That means level 0 is good for those kind of candidates, because we are going to predict the blood and brain concentration here. My drug should go and cross that particular barrier, hence it should be lipophilic, right? So we have various levels here. Then coming to the cytochrome P450. Okay, so this particular enzyme is mostly involved in the metabolic processes, right? So of course, I don't want my drug to go and inhibit that particular kind of metabolism. But of course, it is user defined because some are going to study for the inhibition of this particular enzyme in the metabolic processes, right? So if I'm looking that my drug should not go and inhibit this enzyme, then the level should be false. Okay, otherwise it is true. Hepatotoxicity, which is associated with the liver toxicity. Most of the drugs will show the liver toxicity. Okay, that's why we are checking that kind of criteria or the parameter here, keeping level false. Okay, then once you have taken a drug with the oral this administration, okay, it should go to. That's why we are checking here the human intestinal absorption. This parameter also work well with the when it comes to the prediction of polar surface area, which helps in the drug transform what I would say drug transportation during means or the penetration within the cell membrane. That's why we have kept here this particular range, that is 140 angstrom. It should not be less than that 140 angstrom, right? And we have kept level 0, which means it's good to go ahead, right? The last parameter is the plasma protein binding, which is very important one, I would say. Like when we are going and checking the efficacy of my drug candidate, it should not go and bind to the plasma proteins, right? If it is going and binding to the plasma proteins, that means it is going to the targeted sites. Okay, that means it is reducing the efficacy and of course the bioavailability of my those candidates. That's why if my drug candidate is showing true, that means they are going or inhibiting these plasma protein, that means those are not the good candidate, hence we have kept all those in the level. You can see here under Discovery Studio, we have different protocols where under small molecules, we have these ADMET and the TopCats, right? TopCat is the unique feature of the BioWare Discovery Studio. Okay, once we have done the ADMET prediction for all of the ligand molecule in the series, you can see here after we have done with the filtration, it's easy for me to identify these ligand molecules. I will show you during my demonstration how it would look like. Okay, then coming to the toxicity prediction, that means qualitative, it works with the qualitative structure toxicity relationship. You might have heard about the QSR, right? They're the structure relationship, activity relationship, basically, that work with the some kind of descriptors or the molecular features or those chemical features associated with the structure. Here it behaves with the toxicity relationship, right? We have different various 12 models in the BioWare Discovery Studio. I will show you. We have different aquatic models, aerobic models, we have skin models, we have rat and the mouse models, carcinogenic models, we have mutagenicity. You can identify with the help of these accurate, I would say accurate, because we are getting the data from the FDA, NTP, and the NTC kind of organizations, which are collaborated with us here for those kind of toxicity predictions. Coming to the QSAR, as this terminology is also very common while going for the drug designing process, as I explained that it is the quantitative structure activity relationship. Hence, what we are doing here is we are taking a chemical structure of a molecule and considering those parameters structurally, what are the structure features are involving, those are responsible for the activity of one kind of a ligand molecule, right? So QSAR, we all know that we are predicting those QSAR by using linear dynamics y is equal to mx plus c, where these variable, or I would say the descriptors, okay, we have some dependent variables, some independent variables, and with the help of these variables randomly, which are picked by our machine learning algorithms and for the QSR model predictions, okay, I would say on the basis of those, we can generate some kind of models, okay, on the basis of their activity or the structure activity relationship. These are the parameters of QSR in the BioWare Discovery Studio. We have robust models to generate all those QSR algorithms. You can see we have these 1D, 2D, 3D, descriptors, okay, which work very well with the fingerprint algorithms. We have various fingerprint algorithms which are contributing towards the calculation of, generating some kind of a fingerprints. Actually, I'm not going into that much of details how the fingerprint pin is calculating with some and then it is assigning those models, right? So it is just for your information that we have these various models for the development of KOSAR, where we everyone heard about this MLR, that is the multiple linear regression models, that is the Computational language models. These three models we have in the BioWare Discovery Studio, that is the genetic functional approximation, where the Bison models, we have recursive partitioning models for the development of our QSAR models. Here you can utilize any kind of data, like if you have the continuous data, you can use those data. If you have some kind of binary data on the basis of categorical, like yes or no or active or inactive, those kind of categorical data also you can use here to for the identification of your means for the prediction of your QSAR models. Okay, this Bison model is little bit slow and of course easy to interrupt on the basis of those on the basis of the numerical data you can use. You can handle the fingerprints. Basically, those three work very well with the fingerprints as compared to these two methods. When we are talking about the KOSR, we have to use training set and the test set. We have a different tool for generation of these training set, that is the training set and the test set, where you can see the split method. We have 3 to 4 split methods here, but so it is suggestible to use the random splitting method, because usually those For example, if I'm taking a random percentage, I will choose the another one. Of course, we have some kind of a percentage here. By default, it's set to be 80. That means 80 to 20 ratio. 80 is in the training and 20 could be in the test set. But you can change these training sets. So it's all parameters are user specified to generate all those kind of training sets. Now, coming to the student confusion with the training set, let me tell you, training set is nothing but we are going to train our module or the algorithms, okay, means those molecules which we are using to train something, those are called as a training set. Whereas the test set is when we are going to validate our training set result. We are using QSR, we are using QSR is how it works, what kind of methods are there. Now, what is the applicability of my QSR model? Once we have generated all those QSR models, it's not like every QSR model is good enough or the trustworthy to go ahead for the development of your drug candidate. Of course not. We have to see about those QSR models, which here we are getting those fingerprints with respect to each and every fingerprints of your based on your structures. Basically, we have defined some optimum predicted space, whether if my if my predicted values coming within this particular should be considered as a trustworthy, otherwise not. So those kind of criterias and applicability we are taking care of for the QSR model predictions. Now, coming to the next terminology and the important in the drug designing technique, that is the pharmacophore. Pharmacophore is nothing but it's a structural feature of, I would say, it is an ensemble of your steric or the electrostatic features together to generates or to which are responsible for the biological activity towards a protein structure. As you can see here, those are the complementary feature. For example, if I'm taking a very basic example of a pen, right? So in a pen, what is the main requirement? That is the tip and the refill, right? So that means the design of your pen could be anything, right? So if I'm taking that refill or a tip of that particular pen as a pharmacophoric feature, that means it's an essential requirement, because without those two criteria you cannot define or you cannot have that particular pen as your you can consider as a pen, right? There likewise we have those pharmacophoric features, those are very much essential for its pharmacological activity or the biological activity with respect to any kind of a protein structure. Okay, here within the discovery studio we are using the catalyst algorithms for all those pharmacokinetic pharmacophoric tasks basically. Now, how these pharmacophores will help us, for the development of new candidates. Of course, they are, explaining us about the SAR structure of our ligand molecule in terms of 3D prediction when it comes to the predicting models. Okay, qualitative or quantitative. I'm having the next slide to explain all those parameters. How we can prioritize or select our drug molecule or the ligand molecule from the series on the basis of those active molecules or without completely on the basis of their structure features. We can generate the pharmacophore basically. We can do the scaffold hopping as well for the generation of pharmacophores. Basically, those pharmacophores could be your building blocks, I would say, for the development of your new candidates. Okay, you can see here. As I told you, of course, the docking studies, drug designing is also divided into two categories, ligand based and the structure based. Of course, the pharmacophore modeling types are also like ligand based and the structure based. Ligand based is further divided into two, like qualitative and quantitative. Qualitative is what? Qualitative is basically when you only define those or predict those models without any any quantity. Quantity means here the biological terms, right? Without any biological data, when you don't have any biological activity with you, then you can, then also you can use those kind of ligand molecules which comes under the qualitative, right? We have the autopharmacophore generation, chromophore generation, which works very well with the hip hop and hip hop refined algorithms. Of course, that's why they work with the common feature hypothesis. The other way around, we have the quantitative means we are going to have some kind of quantities means in terms of biological activity, right? So here we can see that we have hypogen and hypogen refinement algorithms in the biodiscovery studio, and it works well with the SAR prediction hypothesis. Okay, when it comes to the structure base, that means here we are utilizing the protein cavity, that protein structure for the generation of our some kind of interaction with the ligand molecule. We can use here the interaction generation for the pharmacophore generation by utilizing some kind of in Ludi interaction maps. Now coming to the docking, as we all heard about protein docking, protein ligand docking. So what are these docking categories? Okay, so protein docking, as its name itself indicates, there are two macromolecules. Okay, both are rigid actually. Okay, they are producing some kind of interaction with no conformation changes. But yes, of course, you can see those kind of changes here as well. Okay, the other way around, those are the protein ligand docking, where the ligand molecule is flexible and your protein structure is rigid. Here you can find out the interaction in between the protein macromolecule and the ligand molecule, right? Now, if I'm taking the protein ligand drug designing itself, it is further divided into two categories, like structure based, as I told you, and the ligand based, where we can going to utilize the ligand structure, right? So in the direct process, like we are having the ligand with us, and we are going to search a protein with respect to that particular ligand. Whereas in structure based drug designing, it's a direct one. For example, means we have a protein structure and we are utilizing that binding site or the inhibitor itself, which is already present in the protein structure for the development of our new candidate, right? Now, what could be the docking flow? Right, we are learned about the protein structure, ligand structure. We have some knowledge about the docking. That what could be the workflow? Of course, we need some kind of inputs for the generation of those to study the interaction in between the protein and the ligand, or the protein and the protein, right? So those are the inputs, like your protein and the ligands. Then of course we are going to check their interaction on the basis of some docking protocols, or I would say some algorithms. I will show you what are the algorithms in the biodiscovery studio. Then of course, on the basis of some kind of scoring function, we are going to select our best poses among all of the list ligand list which we have with us. We are going to filter, of course, the active ones or the good poses. Okay, we are going to refine all those poses, and of course we will be analyzing our results. The validation part is very important in each and every step involved in the drug designing, to make sure, okay, what we have done till now is accurate or with the validated. So yes, this validation point is the most important one. As I told you, we need some kind of inputs. Okay, why we need there is a need of preparation of your protein structure and the ligand structure. As I told you initially, that we are going to mimic our biological system with the in silico ones. That's why in the biological system everything is perfect, having proper charge, hydrogen bonds, some kind of interaction they are showing, some 3D coordinates they are having, no duplicates are there. But here in silico prediction, when we are doing, we need to be take care of all those criteria. That's why we are going to prepare our inputs. So under Biodiscovery Studio, we have one tool to check the protein report. Basically, when you have a protein with this interface, you can very well go and check for its detail, like what kind of crystallographic structure it is having, how many resolution, with what kind of amino acids in the series, what is the pH of that particular protein, what are the chains involved into it, even the active sites prediction means you can identify those active sites from the protein reports itself, whether the water molecules are involved into it or not in the active sites. Okay, so these are the parameters I will show you while doing demonstration. So we have looper algorithm here to fix out all those missing loops. Here with the squares data, it's working well. So it works with the charms force field. That is a very accurate and fast one. Now, once we have protein with us, where to dock our ligand molecule? That is the question, right? So, for that identification of binding site, we have various tools in the Discovery Studio. Like you can see here, protein, from the protein cavities, from the PDB site, and from the current location. For example, I have taken this particular protein structure and it's already having one kind of inhibitor into it. Okay, I can click that particular inhibitor and I can generate the binding site over it with this from current selection, right? So that's how we are generating the binding site with the various of binding site algorithms. I think in my upcoming slide, yes. So you can generate the sites on the basis of protein shape, okay, and of course on the basis of a dock ligand into it. This in the under Discovery Studio, we are searching for the sites on the basis. We have some site search algorithm where the protein is, keeping into the grid, and those algorithms are searching for the free point and what you can see here when it is showing the red in the protein points, and these are the free points available. It will start from one side, it will erasing all these free space to find out the cavities within this particular protein structure. And of course it's work well. Once you have identified those flood filling algorithms identify the possible binding sites with your protein structure, those are fast and good enough to have those for the identification of binding sites. You can utilize any kind of a protein shape, so there is no restriction about the orientation of the protein structure with while keeping into the grids. Once you have done with all your proteins, we have another tools that called as a clean protein. For example, if any kind of functionality is missing or the bonds, amino acids, any false as all it's all after doing all those something is there, you can very well go and clean your protein with the help of these tools here in the Discovery Studio. Now, that is the part with the protein structure. Now, coming to the ligand structure, right? We can I would say we can filter out or we can do those virtual screening on the basis of these 3 to 5 criteria also. Like we have filter by property. We can filter out our ligand molecule on basis of some kind of a molecular properties. The other way around, we have filter by smart. That is, if I'm saying in the simple terms, it will filter out your ligand molecule on the basis of some kind of a substructures. In simply, right? So you can fix those kind of substructures with respect to your ligand molecules, and you can filter out those ligand molecules as well. The third one is we have filter by the Lipinski rule and the Weber's rule. Okay, so you can filter out those ligand molecules with respect to these rules. We called it as a rule of 5, and Weber's rule will specifically works with the rotatable bonds as with the Lipinski rule. Okay, once you have filtered out your ligand molecules, you need to prepare, of course we need to prepare all those ligand molecules as well. So these are the parameters we have kept for the preparation of ligand molecules. To add the for their ionization, we can generate the two tumors, we can fix the bad balances, even we can remove the duplicates. Of course we are not looking for any kind of duplicates here. Okay, so we can keep those kind of fragments. Once you have prepared the series of your ligand molecules, of course then we can take it further for the studies. Now in the docking, it comes into the picture when you have your protein structure and the ligand structure, and it is very well explained. You can see with this picture that likewise only one lock is having one kind of a key, and that particular key is responsible to unlock that particular lock, right? Likewise, we have a ligand molecule with us. Only that particular ligand molecule based on its conformations, it will go and bind with your protein structure to unlock those activity, or means vice versa if you are looking for the simulation or the inhibition. So that particular ligand molecule will go and show some kind of interaction with respect to your macromolecule. Okay, so here it is showing we have various algorithms or the methods of doing the docking in the BioWare Discovery Studio, like LibDoc, Gold, C-Docker, and the flexible docking process. We have, I think, in my upcoming slide, yes, I have explained little, I have given some kind of little description only for you guys. Like LibDoc, it will work very well with the hotspot docking, right? Where it will rank the ligand molecule on the basis of their conformationals. Okay, so it is uses BFG algorithm here, which works, it will generate some kind of polar hotspot and the apolar hotspot. So all your unconstrained non-polar after non-linear optimizations you can do here. Gold is it's basically interface here for the gold. So it works with the genetic optimization for the ligand docking. Of course, it is using the genetic algorithm for the virtual screening, and the for multiple scoring function you can use this gold. Whereas we have the C Docker as well. The C Docker is using the charm force field for the molecular dynamic docking. Basically, it is utilizing a grid-based molecular, means we can call it as a grid-based molecular docking. Whence the charm is involved actually. So it will work with the high temperature molecular dynamics. Okay, when the high temperature is involved into the picture, that means it will, generate some kind of kinetic energies to the system. Okay, in C Docker we can have a ligand as a flexible and the protein as a, rigid molecule. We consider it as highly accurate because it is using charm force field here, those kind of criteria. Of course, we are no need of any prior information about your binding site is required while doing these C dockers, means docking protocols. We have flexible ones where you can keep any macromolecule or the ligand molecule flexible. We have multiple copies simultaneously, such where you can generate the fragments and you can utilize those fragments for the development of your novel candidates by expanding those fragments within the binding site itself. Okay, we have some ligand fit algorithms as well, which works well with the shape based docking with the Monte Carlo conformation samplings. So during demonstration, I will be showing these C Docker ones. Like we need to generate ligand conformations, then the random rotation. It will, on the basis of those random rotations, it will generate some kind of grid-based simulated annealing. Then the further for the minimization will take for the minimization, and of course we are getting output with respect to our, means in terms of our ligand poses. Okay. So here you can see we have those uh, tools to do those kind of docking studies. I will show you. These are the parameters of C dockers. What we have, like if the binding site is particularly large. If, for example, if you're having those large binding sites and you need to explore the further parts of your binding site. So here we have initially C docker is keeping that particular ligand into a grid and generating some kind of spheres here. You can see here. Okay, if the ligand are particularly flexible, of course, and increasing the exploration of your docking orientation, that we have some random or conformations here, and of course you can refine those orientations as well. If your ligand molecule means that those ligand molecules based on their chemistry, maybe they can have the partial charges. Okay, so on the basis of those partial charges, we have ligand partial charge method as well into the parameters of C docker. If the ligand molecule, for example, I am looking for a particular docking of a ligand with respect to my protein structure and it is going to fail. Okay, so I need to increase or decrease those kind of energy threshold values. So we have those orientation van der Waal energy thresholds to to go ahead or give an hit and trial with respect to these threshold values. Of course, we have some post cluster radius, which if I'm looking for with the different poses, so I can increase those kind of post cluster radius as well. So here we are getting the result in terms of minus C docker energy and minus C docker interaction energy. Okay, where this minus C docker energy is the it's energy of your internal ligand strain, I would say. and the receptor ligand interaction, whereas the minus C Docker interaction energy is the interaction energy with respect to your complex. We are getting the results in form of, you can analyze those results with respect to your 2D data, means 2D diagram. Of course, we are getting those interactions. We are getting some kind of tables which are showing the amino acids of your dependent protein interaction or the interface with some kind of a distance. Even we are getting those categories, what exactly this interaction is all about, and the type of that particular interaction. Coming to the molecular dynamic simulation that we called as the MD simulation. In simple terms, if I am telling to you what these simulation is what, it's a movement, right? Simulation is what, it's a movement of, here if I'm considering those with respect to my protein or the complex, here we are considering those movements, or we are seeing those movements with respect to each and every atom in the real time environment. Okay, because we are providing some kind of temperature, we are providing some kind of temperature, we are equilibrate all those temperature to the system so that we can get a final frame into the production zone, right? Because what, why we are doing this, because we are checking what exactly how my complex will behave in the real time environment. Okay, because in the biological system we have the water molecule around every each and every receptor, because maximum 75% is the water in the biological system. Hence we are making those biological conditions here. That's why we are providing all those parameters to the complex to check its stability or the transformations, right? We have various tools we have to analyze the means force field selection, constraint trajectory analysis we can do. We have the protocols like minimization, discrete molecular dynamics, and we can trajectory analysis to analyze our trajectories. Molecular dynamic in the biodiscovery studio, we have quantum mechanics and the molecular mechanics. Quantum mechanics will be taking care of all those bond breakings and the formations, okay, for the small molecules, whereas molecular mechanics will be taking care of atoms, okay. There, your macromolecule could be a larger one as well, okay. It will taking care of the bonded and the non-bonded here. We have various force fields. Now, why there is a force field need to apply while going for the simulation? You must have heard about those force fields need to be applied before going for the simulation. Nothing, but we are making sure that each and every atoms are present in my complex, if I'm telling in the simple terms, right? Because after going to the simulation, okay, for example, if my each and every atom is not proper oriented, then of course it will show some kind of faults to my results, right? That's why we are making sure while applying these force field to the system, so that I am getting a means those kind of charges are proper, those atoms are proper into in terms of their force field. We have various force fields in the biodiscovery studio, as you can see here, okay. But this charm, actually this particular charm, which using the partial charges, Momni and the Rone partial charges, it will take care of all the atoms, whether it is present in the proteins, peptides, or the smaller molecule, with the accuracy, of course. We had different solvation methods like explicit and the implicit. I'm not going into much details of these models, but yes, of course, we are providing some kind of water environment to a particular complex or a protein. That's why we are using the explicit and the implicit, right? Explicit, when it comes, we are going to add some kind of solvent boundaries to it, whereas implicit is a simple terms. It works very well with the continuum dielectric constant, where we have kept it as an 80 for the water permeability, right? So we have different various, I would say the various models where these GBMV and PBSA methods are very popular in terms of their accuracy. Okay. We have robust minimization methods in the biodiscovery studio, that is the first order and the second order. When it comes to the first order, we have the other one also, but I have explained these two here. The steep and descent and the conjugate gradient. Actually, this is steep and descent we call as a first step minimization method as compared to the conjugate gradient, as this conjugate gradient is taking some kind of a history of the steep and descent. I will show you while demonstration. The other way around, we have a second order method like adopted basis set Newton-Raphson method, which works very well with the Newton's Raphson algorithms, which we are, when we are applying some kind of coordinates to the dynamic simulation. These are the tools of Discovery Studio. In under the simulation, you can see the minimizations. We are going there is an iterative process of including the two minimization stages, heating, equilibration, and the production in the final step. Of course, we have dynamic NAMD as well in the GPU and the CPU modes both. So this is the basic, I would say, the workflow of molecular dynamic simulation, where we are going to, put some kind of inputs, okay, whether it is a complex or protein or a protein ligand. We are going to apply some kind of force field. Then we need to apply the solid state by providing some kind of, ions. Then we are proceeding with the iterative step. Finally, we are analyzing the trajectories. So these are the parameters of Biodiscovery Studio for MDS. First dynamic step, minimization step. The other one is taking the conjugate gradient with some RMS gradient. Then the heating, here we are going to save each and every result after heating. Again, some other calculations are here if you are going further for the higher, molecular dynamics for the nanoseconds. Here is the equilibration. This heating results will go directly to the equilibration to make sure whether the system will equilibrate with respect to the heating or not. Then we are going for the production finally, where it is using the type NPT, that is the number of atoms at constant pressure and temperature. We have two more. I will show you while doing demonstration. Here we have dynamic integrator with us, that is the leapfrog wallet integrator, which works very well with the shake constraint. This is the parameters for the NAM dynamics. So you can see the simulation times, whatever you are, a user is looking, keeping their simulation with respect. Here it is mentioning in the picoseconds. Okay, we have various platforms like GPU modes and the CPU modes. You can restart your dynamics while putting those restart files which generated during the this dynamics in the Discovery Studio for extension of your dynamics further. Here also it is taking the modes that like NPT. We have two algorithms like Langevin dynamic algorithms for the temperature control, whereas Langevin piston algorithm for the pressure controls. Of course, we have a variety of analysis tools and the protocols with us to analyze all those kind of pharmacodynamic results. Likewise, we are getting results when we are comparing two complexes, like this particular complex is showing RMSD. RMSD is nothing but it's a root mean deviation. Okay, how it is deviated from its starting means frame. As compared to this one, then this is the RMSF, that is the root mean square fluctuation. They are studying for each and every atom here. Okay, you can see here this is more fluctuated as compared to the other one. At the radius of gyration, we are looking for the compactness of your complex. Okay, so what could be the conclusion if I am concluding all those criterias? Means if I am looking for the conclusion, we have used QSAR models, pharmacophoric models, docking studies, virtual screening, ADMET properties. So all those criteria, why we have used, and what are the outcome from using all those kind of models and the predictions, right? Of course, it increase your efficiency and reduce the cost. Of course, we can identify the targets and we can validate those targets by using all those kind of algorithms. Okay, when it coming to the means when it coming to the identification of those identification we can do with the help of structure based algorithms or the pharmacophoric algorithms when it coming to the how we can optimize those kind of optimize our ligand molecule. Of course, we can use, as we have seen during the different sessions, we can reduce the animal models. Even we can go for the personalized medication by using all those kind of algorithms by improving the advanced algorithms. We can drive our data from huge libraries to the when it comes to the filter out all those ligand molecules. Okay, as this drug designing process is not a one step process, it's an iterative process where ligand molecule filter out steps are involved and the QSR pharmacophoric models predictions are involved on the basis of some kind of fit values. Then the docking is involved on the basis of some kind of functionality in terms of scores, so that we can select all those good poses for the further studies of the molecular dynamic simulation to check the stability of that particular complex and the macromolecules. And of course we are mitigating the risk factors what we are not in the conventional part, right? So here is a list of a very few successful drug with the in silico, right? We have some HIVs and all those kind of drugs, but of course how we can forget about the SARS-CoV-2. Recently we have got those kind of vaccines. Even the biovid, I think I have the next slide, which showing its capabilities, where we are having some antibody designing tools, even the pharmacophoric tools, which are contributing towards to the generation of all those kind of vaccine developments kind of process, where the simulation, okay, we starting from the homology modeling, protein docking, all those kind of modules we can utilize under the macromolecule. Then downstream we can do the virtual screening, we can go for the analysis, we can go for the QSR and the toxicity predictions. We can develop the scaffold hooping. We can replace those fragments within the pocket of the protein structure itself, so that we can profile all those ligand molecules. And of course ultimately we are going to check the interaction of those macromolecule with respect to the ligand molecule. Okay, so I can say that okay, this discover finally the because we are dealing with the bioinformatics and the chem informatics here. That's why I could say that biodiscovery studio itself is a solution for when it comes to the bioinformatics and the chem informatics. Let me take you through the demonstration. Just give me a minute. Hmm. As you can see here, this is the biodiscovery studio software. Okay, so let me take you first in the drive of how we can upload the series of the ligand molecule. If I'm opening any kind of you know like PubChem. Let me share my screen again. Just a minute. So hope you are able to hear me. Is the come bell? as a database. Meanwhile, let me take you to the Discovery Studios, this software interface. So you can see we have the various modules tab here. We have some menu bars here. This is the molecular window we have. We have various jobs window and the help window here. We have tools. We have protocols for each and every modules actually. So, for example, if I need to go for QCR studies for some of the ligand molecule, let me take some kind of a ligand molecule. Okay, so you can see these. Here is a list of, for example, how many we have 3,000 ligand molecules. So I need to select those. I need to filter out all those ligand molecules on the basis of their structures. I mean, ADMET and the toxicity prediction. How to go for these kind of filtrations? Let me go. Let me run first for the ADMET prediction. Go to the protocol. Let me show you if I want to save this data to one kind of a folder. I have already saved it. You can see this set as a default. By clicking on this folder will become a default to you. Okay, so whatever you are doing, it will go directly to this particular folder now, right? So go to the protocol. Under small molecules, under ADMET, we have ADMET descriptor. Hope you are able to see my screen. दिख रहा है? नहीं अभी नहीं दिख रहा, जब दिख रहा था. Let me share my screen again. Share screen. Hope this is visible to you. In my Discovery Studio. Hope this is visible. Let me take some samples again, or the structures to study the this QCR studies. Okay, let me take quickly to the protocol under small molecules, ADMET and ADMET descriptors. It will open like this. You can see here, we can see the molecules in the list, like we need to keep it as on. Here you can see we have the various parameters to calculate ADMET. After selecting all those ligand molecules, you can just give a run to it. Let it run for a while. Okay, I will show you how these results will look like. So it will ask you about to open the ADMET plot. Okay, it has calculated the ADMET property for 3007 molecules. After seeing this particular graph, I am not able to, identify what molecule is good enough to take further for the toxicity and the docking and the further the MDS simulation studies. Even if you want to take the molecule you can go and you can open it. We have further color identification for each and every UPS area. So you can identify it from here also by seeing the colors. But of course, I'm going to filter all those 3000 molecules with the help of those level filtration. Okay. So for example, if I'm taking the ADMT solubility levels, I'm keeping this equals to 3. Okay, then BB level, you can give these levels one by one, or you can go for simultaneously. Then further for this CYP prediction equals to false, of course. Then for the hepatitis absorption level, I'm keeping it as a 0, and on this plasma proteins equals to false, and just give a apply to it. You can see here how beautifully it has cut down to fused ligand molecules, and now I am able to see those ligand molecules within the OPS area. Even we have we are getting some kind of estimated values with respect to each parameter here. Okay, you can see for each and every parameter we have predicted values. And how much ligand molecules we have got? We have got 60 ligand molecules from the 3000 ligand molecules from the series. Okay, let me take these molecules further for the toxicity prediction. If you want to save this particular window, you can go to the file, save as. It will automatically open to your in this folder only, and you can give it as ADMET. Okay, and you can save it. Now further I will be taking this to the top get prediction. Okay, I need to give these all. Okay, so you can see if you want to add some further models to the prediction, you can click and you can add. So you can click and you can add. So I'm not going to take every model here. Okay, just let me take the one and give a OK. It will generate those data on the basis of some bad contribution and the positive contribution with respect to the chemical structure, and it will generate a detailed report and giving in the PDF format. Okay, just let me, after setting these parameters, you need to give a run to it. It will start this job. Let me show you how the results will look like after doing these toxicity predictions. We are getting some output of the ligand molecule, and here we can see the detailed report with respect to each and every model what we have given to the algorithms, which works very well with the Bayesian score. You can see here it has generated some molecular properties with respect to this particular chemical structure, and of course it has done the similarity search, and it is providing us the positive contribution towards the toxicity, and of course the negative contribution towards toxicity for each and every ligand molecule. If you are giving the two models, whether you are giving the one model, it will generate a PDF format report for your toxicity predictions, right? So this is about the ADMET and the toxicity. Now let me take these models. Okay, not these. Let me take models for the docking protocols. Okay, we have receptor ligand interaction. Of course, for docking, we need some protein structure and we need some kind of a ligand molecule, right? So let me download that protein structure for you. For example, if I'm taking this only, okay, this is a protein structure. As I can show you the protein report. This is the protein report, okay, where we are getting the information about the crystallographic structure, its ligand, what is the active site, what kind of. Here you can see this active site involves both the chains, right, A and B, with some water molecules, right. So let me show you how to select those, even the PDB information with the name of this particular proteins we are getting. Okay, this is the chain, this is the chain, this is the ligand, and this one is the payload. So we have those water molecules which are intact in the active site. By clicking, just shift and click all those, selecting these ligand molecules. Let me do it again and delete. Okay, the from the other on this chain as well. Delete, delete. Okay, so now this is the protein is prepared using the tool, that is the preparation or prepare your protein. Click on this. You can see we have the build to square from square data looper proteinase. It will generate some kind of advancement, but it's taking the charm force fill. I need to keep those water molecule intact. Okay, so let me select all and give a run to prepare your protein structure. Let me show you quickly. Once your protein is prepared, okay, you can take this particular protein. Let me close all those windows. Okay, here you can explain this. You have this particular ligand molecule, this one, and you can generate those binding site if you don't have any information about those binding sites from the interaction. Define, you can select. Okay, even you can expand this your binding site, or you can contract it. Okay, after assigning those binding site, if you want to do, I mean, we need to assign it in a way so that our molecule will go and bind to that particular site. Let me. Okay, and final step is that we are going to have those ligand molecules, and we are going to dock. So let me take that protocol, cdocker, as it is open, my ligand molecules. Let me take very few, means one or two molecule for the cdocker. Let me take two. Okay, so these are the receptor paired one. It automatically generates the site sphere for me. I'm not taking all, I'm only taking visible one. Okay, hotspots with some cluster radius, dynamic steps. Okay, I'm not touching all these parameters. Okay, I just give a run to it. Let it run. Let me show you the results from the topic. While clicking this view results, it will open the results and showing these table with respect to the, my ligand molecules, my Docker interaction and energy, of course, some poses, and those are the ADMET results what we have. Okay, while with the help of these arrows, you can select the ligand. Need to go to the receptor ligand tools, view the interaction, a ligand interaction. Okay, so likewise you can predict the interaction of your ligand molecules with respect to the receptor. Even you can show the distance, okay, types as well. If you want visualization of the interaction, you can do this one as well. So likewise we can generate non-bonding interaction details also in our tabular form. So this is what I'm having for today's session. So we can go for the virtual screening, how we can utilize those algorithms to do the virtual screening, and further for the preparation of our ligand molecules and the protein molecule, then how we can utilize those algorithms for the to check the interaction, and of course many more in the molecular dynamics and the binding energies. So that is what I'm having for today's session. So if you have any questions, you can let me know.
Thank you, ma'am, for such a nice talk. Now, this is the time to propose vote of thanks on behalf of Department of Pharmaceutical Chemistry, Faculty of Pharmacy, Swami Vivekanand Subharati University, Meerut. It gives me an immense pleasure to deliver vote of thanks for this webinar on advances in computer-aided drug design from algorithm to therapeutics. First of all, I would like to thank our Honorable Vice Chancellor, Major General Dr. G.K. Tapalyal sir, and Chief Executive Officer Ma'am, Professor Dr. Salya Raj, for providing us support and facility to conduct this webinar successfully. I would like to thank our speaker ma'am, Dr. Sakshi Bharadwaj, application specialist, life science, Altem Technology Private Limited, who despite her busy schedule has found the time to grace this online event by deliver lecture. I would like to thank our principal, Professor Dr. Sukhinder Kumar ji, Head of Pharmaceutical Department, Dr. Ganesh Prasad Mishra ji, faculty members, students, and participants for making this webinar successful. Once again, I thanks one and all present here. Jai Hind. Thank you, ma'am, for your presence.
Thank you so much, sir. If you have any kind of questions, you can even mail us. You can write the mail to us. We will be,
Sure,
answering your questions there.
sure, ma'am.
Thank you so much.
Thank you, ma'am. Thank you.